Hello everyone, this is Mr. Schultz, and this is the AP Biology video covering the cell. This is video 2.1, so this is a new unit, and this video covers Biology in Focus Chapter 4. Unfortunately, this video is probably going to be pretty long because there's a lot of material to cover and it's an entire chapter instead of just a small section of it. Also, please be aware that there is a part of this textbook that covers the microscopes. We won't go through those in the video today. If you have time, please make sure you read through that section of the textbook, please. Now, let's talk about the cell. Now, all cells have certain things in common with them. So one of the things that all cells have is what's called cytosol. It's a jelly-like fluid in all cells. When you're in middle school, a lot of times we talk about like the cytoplasm as being like this liquid inside of the cells and it sloshes around, but it actually is more gel-like than anything else. And the interior of the cell itself is actually called the cytoplasm. So just be aware of that. The cytosol is actually the liquid itself. Now, all cells contain chromosomes. Depending on the type of cell, you might have a single loop of chromosome, one DNA strand that basically is one big loop. Bacteria and archaea are like this. Or you might have multiple chromosomes. So like an example would be human cells. We have 46 chromosomes, 23 from mom and 23 from dad. Each of these chromosomes is one long string that's basically bundled together. But keep in mind that bacteria usually have just a single chromosome. And they might have what's called a plastid, which is a tiny little bit of DNA as well, which is used for transferring genes and new traits between bacteria and things like that. All cells also have ribosomes, which are used for making proteins, and they all have a plasma membrane. Now, the actual plasma membrane itself might differ slightly in structure and the proteins found on it, depending on the cell, which would make sense because there's a lot of different cells and there's a lot of different proteins, particularly identification proteins that help keep the cells both safe or identify it to other cells around it. So, when we talk about cells in and of themselves, there are two big groups of cells. There are prokaryotes and eukaryotes. Pro means before, and karyo means kernel. So when scientists were first studying the cells, they looked like kernels or small pieces. So prokaryotes are things like bacteria and archaea. Their DNA is concentrated in what's called a nucleoid. It's not a protected organelle. It's just like a clump of DNA. There are no membrane-bound organelles, which is a big deal. There are no mitochondria, there's no Golgi, there's none of that stuff. So they're very basic. Prokaryotes are also much smaller than eukaryotes. They're usually 1 to 5 micrometers wide or big. So they're much smaller than eukaryotes. Because eukaryotes, they have a nucleus, which helps protect their DNA from things like viruses and other stuff. And it also helps kind of control like protein synthesis and stuff as well. They have membrane-bound organelles. They have mitochondria. They have chloroplasts if their plants are protists. Some protists, not all. They have a Golgi. They have endoplasmic reticulum. They have lysosomes in animal cells anyway. Vacuoles. And they're much larger than prokaryotes. So they're usually between 10 and 100 micrometers. What's really interesting about eukaryotic cells is they can be much bigger. But like if you think about our nerve cells, they can be really, really long, but they're very, very thin. So they're still relatively small speaking, but they're just very, very stretched. And that brings us to a really important question in biology, and that's why can't we have giant cells? If we look at like a blue whale, it's not that it has huge cells, it just has many, many more cells. So cell size is dependent on the surface area to volume ratio, and we're going to do an activity in class to study this. So if the math is still a little tricky, don't be too worried about it because we'll go more in detail in class about this. So as a cell grows larger, the volume grows more than the actual surface does. So the volume inside the cell gets much bigger, much faster than the actual outside skin of the cell, the plasma membrane. When we have more surface area, it means we have more ability to let things leave and enter the cell. So cell transport is very much dependent on how much plasma membrane there is. And so if you're really, really big, let's say we have a cell like this big, it would take forever from like an oxygen molecule to go from outside the cell to inside the cell. And that would be a problem for the cell to live. We can't maintain homeostasis if there's just no movement or it's very slow. So smaller cells are better than bigger cells for the most part. Some cells are very long, like our nerve cells, but they're usually very, very thin. And what happens is, if you think about it, you're pulling it and stretching it, which increases the surface area. So it could be a relatively large cell in terms of length, but it's actually got a very large amount of surface area, and there's not a lot of movement that has to happen for oxygen to go inside the cell. And some cells are maximizing or will maximize their surface area for absorbing nutrients. So an example of this would be the cells inside of your small intestines where they're absorbing a lot of material. They're going to want to increase their surface area so they can absorb as much stuff as possible so that it can be modified, shipped, moved into the bloodstream, wherever it needs to go. If you're interested in looking at the math behind this before we get to it in class, you can look up on page 74 of our textbook. There's a quick math activity I would highly recommend you try because it's very similar to what you might see on the AP test. When you see those little calculation activities, they're a good idea to try. So let's talk about all the parts of the cell. This is where this video gets really, really long because there's a lot of parts. And you do actually need to know all of them. Because they could come up in AP test questions. It could be like, hey, what are 3 organelles that you would expect to find in this type of cell? Or it could be, hey, something went wrong with this organelle. What could happen afterwards? So just be aware of that. So the nucleus contains most of the genes of the cell. The reason we say most is because some organelles in the eukaryote will also have their own DNA. The nucleus itself is surrounded by what's called a nuclear envelope. It's actually a double membrane with a whole bunch of pores interspersed across it. And the purpose of the pores is so that it can allow for mRNA to leave the nucleus. The nuclear envelope also has what's called a nuclear lamina, which is like an inside thin layer. The chromosomes, they're just one long DNA molecule, and depending on what type of organism you are, you might have a lot of chromosomes or you might only have a few. Chromatin is the DNA and the proteins that hold the chromosome together. So the chromosomes are the actual DNA. Chromatin is DNA and the proteins that hold it together into a structure. And there's a whole bunch of information that goes with that we're not going to get into in this unit. We'll talk about more when we talk about DNA as a molecule and a means of getting information. The nucleolus is a small interior part of the nucleus that helps assemble ribosomes. And we like to think of the nucleus as controls the cell activity simply by controlling the protein synthesis. It controls which parts of the DNA get read and transcribed into mRNA. So the nucleus controls the cell by controlling how proteins and when proteins are made. So be aware of that. We always talk about it as like middle school, like, oh, the nucleus controls the cell. Well, this is how it does it. It controls the proteins. The ribosomes, which are found in both prokaryotes and eukaryotes, are made up of ribosomal RNA and protein. So it's a complex of protein pieces and RNA. And it carries out protein synthesis. And there are 2 types of ribosomes. There are free ribosomes, which are suspended in the cytosol, and they're used for making proteins in the cytosol itself. So like any proteins that are needed for like glycolysis, you would expect to be made by free ribosomes because glycolysis happens in the cytosol. If a protein is going to be attached to something, it's going to be made by a bound ribosome. Bound ribosomes are attached to the endoplasmic reticulum, which we'll talk about in a minute, and they're used for inserting proteins into the membranes themselves. So if you have if you're going to make a new amount of cell membrane to replace maybe one that's starting to struggle or falling apart, you would get your ribosomes from your endoplasmic reticulum to make them within a membrane already, and then it would get modified and shipped. So the ribosomes are sometimes attached to what's called the endomembrane system, or the ER. We have the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, vesicles, and vacuoles are all part of what's called the endomembrane system. They're organelles that are surrounded by a membrane. And you're going to see this word from time to time. It's called a vesicle. It's a sac that's made up of a membrane. So think of it as like a pocket. I always like to think of them as like Ziploc baggies that are just floating around. And depending what's in the Ziploc baggie determines what's going to happen and what the organelle does. So the ER, or endoplasmic reticulum, is directly connected to the nuclear envelope. So it's actually attached right to the nucleus. And the reason for that is it's much easier to get mRNA from the nucleus to the ribosomes if they're right next to each other. There are two types of ER, the smooth ER, which is used for the synthesis of lipids, metabolizing carbs, detox is sometimes done in the smooth ER, and the storage of calcium ions is also done by the ER. There are no ribosomes attached to the ER, the smooth ER, excuse me, but the smooth and rough ER can be attached to each other, so just be aware. They're just like two separate parts of the same thing. The rough ER has ribosomes attached to it. That's why it's called a rough ER. It creates membrane-bound proteins. It can also create what are called glycoproteins, which are proteins that have a carbohydrate, or glyco means sugar, so like a sugar attached to them. The proteins are packaged in transport vesicles, which are then shipped to other organelles, specifically the Golgi. It also will help build the phospholipids for the membrane as well. So just be aware that the rough ER has a number of jobs, most of which revolve around making proteins for the membranes and then helping make the membranes themselves. The Golgi apparatus, which looks like a whole bunch of little like flat sacks that are on top of each other, it's kind of a stack of them. And so they're flattened, and what they do is they modify, they store, and they ship all the products from the endoplasmic reticulum. So if the rough ER makes like a chunk of membrane with a bunch of proteins on it, the Golgi helps like modify it so that it's ready to go and it can be inserted where it needs to. There's a large number of them found in cells that specialize in secretion. So if you think about like mucus secretion in your nose cells that secrete mucus will have a lot of the Golgi apparatus because it helps make the mucus and prepare it for release. So you're going to expect to find more in places that secrete things. So just be aware of that. And they do some synthesis of macromolecules. Sometimes they help with making other larger molecules. But for the most part, they're there to modify proteins and membranes to get them ready for wherever it is that they need to go. Lysosomes are a sack of hydrolytic enzymes that animal cells use to digest macromolecules. They are found in animal cells, so be aware of that. And so what it does is it attaches to an organelle or a big old vesicle full of food product, like big chunks of something, and then it releases these enzymes into the other vesicle, and then the enzymes break the foods down, and now we have digestion. The term that we use for like digesting engulfing food is called phagocytosis. So an amoeba, which is like a really cool protist that can extend itself to eat things, wraps itself around like a blob. Think of it as like a big old piece of slime. And so it engulfs a food particle, or like it could be like another bacteria or something like that. So it wraps itself around a bacteria. Now this bacteria is stuck in a vesicle. And so what the amoeba will do is it'll take a lysosome and it'll attach it to it, and the lysosome will release all these enzymes to break the food down. And then be aware that there's also a term called autophagy, which is the self-digestion. So lysosomes do autophagy for organelles that are dying. So if you have a mitochondria that just isn't doing it anymore, or something happened and it's damaged, a lysosome can attach itself to it and break it down to reuse the materials that were being used for the mitochondria originally. Vacuoles are very large vesicles. Occasionally, you'll find what are called food vacuoles from phagocytosis. So like if an amoeba eats something, we call it a food vacuole. There are also what are called contractile vacuoles, specifically found in certain protists particularly. And their job is they pump excess water out of the cell, and they help control ion concentrations in cells. If you're a cell and you live in a freshwater environment, and you have a lot of nutrients inside of you and a lot of particulates and stuff, we call them solutes, and you live in a freshwater environment, the water is going to want to go into your body to spread that stuff out via diffusion and osmosis. That would be bad for a cell. It would explode. So we don't want that. We want these cells, they use a contractile vacuole to push the water out of the cell, which helps protect it and maintain homeostasis. Another example of a major vacuole you need to be aware of are what are called central vacuoles. And it's a large vacuole in plant cells. and it's filled with inorganic ions and water, and it helps create what's called turgor pressure, which helps hold plants up. So if a plant doesn't get enough water, it starts to get kind of droopy. That's because there's no water pressure helping push against the outside of the cell. So vacuoles are really important, particularly in plant cells. The mitochondria, the powerhouse of the cell, as you've probably heard since like first grade, is the site of cellular respiration. It uses oxygen to make ATP by pulling energy from other molecules. I don't ever want to hear that the mitochondria is the powerhouse of the cell in AP biology. At this point, we should be able to say, hey, the mitochondria takes biomolecules and turns them into ATP via a number of chemical reactions. It consists of two layers. The infoldings are called cristae, and their main purpose is to make a huge amount of surface area. The more surface area that you have, the more work you can do. And we want to do a lot of work because we want to make a lot of ATP. The area within the inner layer is called the mitochondrial matrix, and it's a type of gel. And if we look at a mitochondria, it actually has its own DNA. So in this picture, there's these little loops. They're little DNA loops, and they're actually evidence that mitochondria at one point were probably their own organism. And we'll talk about that in a later video because it's really interesting. They're found in nearly all eukaryotes. It's pretty rare for a eukaryotic cell not to have mitochondria. And the more activity a cell does, the more mitochondria you can expect to find in it. An example of this would be our muscle cells have a very large number of mitochondria because it takes a lot of ATP to contract and release muscle proteins. So you would expect to find way more mitochondria in a cell that does a lot of work than, say, a cell that maybe just sits there and does one specific function where it still needs ATP, a lot of it, just not nearly as much as some cell that's doing a lot of work. So just be aware of that. The more mitochondria, generally the more energy needed by that cell. Chloroplasts are the site of photosynthesis done by plants and algae. So be aware, chloroplasts are found in more than just plant cells. They're also found in protists called algae. They contain the green pigment called chlorophyll. And chlorophyll is green because white light comes down and chlorophyll absorbs all colors but green, basically. And the green reflects and we see green. So be aware that chloroplasts contain interconnected sacs called thylakoids. And a stack of thylakoids, I think of them like a stack of coins, are called a granum. The fluid outside of the thylakoids is called the stroma. The chloroplast itself is going to contain DNA, its own ribosomes, and its own enzymes. Again, another piece of evidence that suggests that chloroplasts, like mitochondria at one point, were their own cells. And then they're part of a group of plants called plastids. Plastids are organelles found in plant cells and algae cells, and they're used for storage or for specific jobs. Other plastids within a plant cell might be an amyloplast, which is used for storing starches, high energy storage for plants. And then you might also find what are called chromoplasts, which are storage pigments for giving fruit and vegetables. There are fruits, vegetables, and flowers. They're yellow and orange colors. So if you have a really bright flower, you would expect to find some chromoplasts with specific pigments in them to attract things like bees. Now, the evolution of mitochondria and chloroplasts is really important. It's one of the key pieces of evidence for evolution as well as for where eukaryotes came from. The endosymbiont theory it claims that an early ancestor of eukaryotic cells engulfed an oxygen-using, non-photosynthetic prokaryotic cell, and so the cell became what's called an endosymbiote. Basically, a bigger cell ate a little cell, the little cell did something cool, and so the cell kept it around, the big cell kept it around. That's pretty important. It allows you to kind of absorb and basically, instead of eating a mitochondria, it kept it around because it made all sorts of energy, and that's really, really cool. We have a number of pieces of evidence for this theory, by the way, because a good theory is going to be backed up by evidence. One, chloroplasts and mitochondria have double membranes instead of one, like normal regular organelles, like an endoplasmic reticulum. They contain their own ribosomes, and they're unique to their actual organelle. They're not the same ribosomes found in the rest of the cell. They contain their own little circular DNA, like a bacteria, and they're somewhat independent from their parent cell. They actually undergo their own version of cellular division, and they do it on their own. And then when it comes time for the cell to split, the mitochondria will actually break into groups and go into separate sides. Another type of organelle found inside of the cell is called the peroxisomes. They're a specialized single membrane compartment, and their job is to help form and break down hydrogen peroxide in the cell. So they contain enzymes that remove hydrogen from certain molecules and transfer them to oxygen. And what they make is hydrogen peroxide, which is technically toxic to the cell in larger quantities. And so what it'll do then is it'll pull hydrogens off of poisons like alcohol in the liver, and then eventually it will use enzymes to break the hydrogen peroxide down into things like water and oxygen, so that the cell can use those products. Because water is just a waste product at this point, and oxygen can be reused in respiration. Now, the cytoskeleton is also a structure that helps, it's a network of fibers that extends throughout the cytoplasm, and it provides mechanical support and provides shape for the cell itself. And on these pieces of the cytoskeleton, you might find things like motor proteins, which aid the cell in moving along, as well as organelles to move inside of the cell. So motor proteins can help move a cell itself and move things inside of it. There are also centrosomes, which are part of the cytoskeleton. It's a microtubule organizing center, and it's going to come into place particularly when looking at mitosis. So the centrioles, which are located in the centrosome, they play a role in mitosis in animal cells. And we'll get to that when we talk about mitosis in another unit. Other pieces of the cytoskeleton you should be aware of are the flagella, which is a whip-like tail found in sperm of animals, algae, and some plants. An example of a plant that would have a sperm is like ferns do this. There's usually only one or a few on a cell, and they're used for locomotion. And there's some really interesting data coming out where originally it was thought that like sperms swim like a fish swims, but now we're finding out that they spin. And if you look at this diagram, flagella, they actually spin in a circle, and that's how the organelle moves, or the organism moves, sorry, not the organelle. Cilia are a whole bunch of tiny hair-like projections, and a cell will have a whole bunch of them. So instead of having like one or two, there's like hundreds. And they can be used for locomotion, for moving and gliding through water. They can also be used to actually move fluid. So if you think about like in our throats, in our lungs, fluids have to move around because if they get caught, then we get sick or we can't breathe. So cilia help move that along. They can also move things like dust and stuff in our nose. They can also be used for signal receiving as well. In this case, you would see a singular cilium. Okay. Microfilaments are called actin and myosin. We're not going to get into them in too much detail. You would study them much more in an anatomy and physiology class when you talk about muscle proteins and how muscles work. If you are interested in learning more about this, I highly recommend looking it up because it is really interesting how our muscles work. In terms of outside the cell, there's what's called the cell wall, and it's found in plant cells. It helps protect the cell, it maintains the shape of the cell, and it prevents excessive water intake, because if the cell didn't have this, anytime it rained, plant cells would just absorb way too much water, and that would be really bad for the cell. It's made mostly of cellulose, which varies in thickness and structure from plant to plant and from cell to cell. So the cellulose in one part of the plant might be much tougher than in another part of the plant. And we like to think of cellulose as being fiber in our food. So if you've ever eaten a high-fiber food, you would expect to find a lot of cellulose in this. In fact, like wood is almost all cellulose. So unless you have bacteria in your gut to break these things down, we can't digest them. And so our body goes, hey, we can't use this, so let's get rid of it. And so we put a lot of mucus around it, and then it helps you push it out. And so that's what fiber does. It helps you poop. So if you're ever wondering why, that's why. Now, the plasmodesmata are these tiny little channels in the cell wall, and they're going to allow water and small solutes to pass between the plant cells. And so they're just kind of a way that water can kind of move back and forth between cells depending on if they need it or not. Also be aware that in animal cells, there's a layer of interconnecting glycoproteins and other carbohydrate containing molecules. The most common is collagen. it's about 40% of human protein actually is collagen. So that's a lot of our protein. And the extracellular matrix, it helps hold the cells in place, because if it didn't, your cells would start wandering and moving around, and that would be really bad. You wouldn't want your heart cells to be wandering off. That would not end well. So the extracellular matrix kind of helps hold them all in place. There's a few other things that we look at between cells, and they're called cellular junctions. Plants, they're going to use their plasmodesmata, but in animal cells, there are 3 types of junctions. There's a tight junction, where plasma membranes are held tight together. That's kind of one of those things where 2 cells are held together really, really tight. An example would be skin cells. The reason our skin cells are relatively waterproof is because tight junctions hold all of our skin cells together. There's the desmosomes, which fasten cells together like rivets, and they're kind of what help hold our muscle cells together. Because if you flex your muscle, what's going to happen is if they don't hold together, your muscles are going to pull apart, and that wouldn't end well. We want our muscles to hold together. And then finally, there are what are called gap junctions. They provide channels for cytoplasm to pass between 2 cells. And they're not, I don't want to say they're not common, but they're not, in terms of like all of our cells, common. An example would be of an actual gap junction is our heart cells have gap junctions because the heart is constantly working together to do one major function. So if they don't have to keep sending signals that are a little bit slower, this allows for quicker signal movement as well as quicker nutrient movement as well. How did we figure all this out? Like, how do we know what the organelles do and what they look like and stuff like that? Well, we use an electron microscope to look at the actual organelles themselves. But before we can do that, we have to do what's called cell fractionation, which is kind of neat. You use a bunch of enzymes to break a cell apart. So you put some cells in a test tube, and then you add the enzymes, and then you spin it really, really fast in a centrifuge. And what happens is the bigger components tend to settle to the bottom, while the smaller pieces stay at the top. And so you get all of your big organelles, and they'll divide up into layers according to what they are. And then once they separate, we can use like a pipette to pull out one layer at a time and put them into the microscope to study them. And so if you wanted to study like mitochondria, you could break a whole cell apart, you could fractionate it, figuring out which layers your mitochondria, pull just that layer out, and now you have a bunch of mitochondria to work with. So this is the end of the cell video. I'm sorry it's really long. Unfortunately, you do need to know all those organelles, and you need to know their functions. So be aware of that. So if you're having a hard time with the organelles, my recommendation is to make flashcards, because there's a lot of them and you need to know them. So our next video will be talking about the plasma membrane, but until then, enjoy the rest of your note taking.